Today, my presentation is on a primary research article entitled Lithium Treatment Reverses Irradiation-Induced Changes in Rodent Neural Progenitors and Rescues Cognition. Just a little background on this research article. The primary researchers are listed, and they had a number of affiliations across the globe, including Japan, Stockholm, South Africa, Brussels, the USA, and Greece. And then it was published in the Journal of Molecular Psychiatry in October 2019. So, a little introduction. There have been dramatic improvements in childhood cancer survival rates, but cranial radiotherapy in children has led to detrimental effects on cognition, such as impaired processing speed, attention and working memory deficits, and declines in academic performance, all which are impacting their general quality of life. There is a particular interest in the neurogenic regions of the brain because they demonstrate a higher sensitivity to irradiation, for example, apoptosis and progressive decline in neurogenesis. Lithium specifically is a common treatment of bipolar disorder, but has also been shown to exert neuroprotective and regenerative effects. But its exact mechanism is only partially known/understood. The research goal of this paper was to investigate the effects of lithium treatment on neural stem progenitor cells, also known as NSPC, and whether therapeutically relevant doses can rescue neurogenesis and ameliorate memory deficits even long after irradiation of the juvenile brain. So the methods included introducing lithium 3 weeks after female mice received a single 4 GY whole brain radiation dose on postnatal day 21. The mice were then randomized to either 0.24% lithium carbonate chow or normal chow from PDN 49 to 77. Then hippocampal neurogenesis was assessed on PDN 77, 91, and 104. The researchers performed immunohistochemistry, dendritic reconstruction and morphometric analyses, protein quantification, RNA, cDNA, RT, and ME-DIP, qPRC, as well as behavioral assessments with the Morris-Water maze. The researchers found a number of things, the first being 4 weeks of continuous lithium treatment increased the number of proliferating cells in the irradiated brains by 83%, but had no effect on the double cordon positive cell density in the granule cell layer. This is indicated in figure E in the top right corner. In addition, lithium discontinuation prevented irradiation-induced alterations in process orientation and dendritic complexity of DCX plus cells on PND91 2 weeks after discontinuation. The DCX plus cell density decrease caused by irradiation was reversed by lithium treatment. In addition, dendritic complexity of DCX plus cells in the irradiated group was restored completely by lithium to the sham level. This is indicated in figure G in the bottom right corner. So the first thing I want to talk about are figures F and G. This refers to how they found lithium upregulated genes that were involved in the cell cycle and neuronal signaling after radiation in an in vitro part of their study. They looked for the highest fold changes in gene expression between sham and sham treated with lithium, as well as between irradiated and irradiated treated with lithium. They highlighted 2 genes, one that encodes for tubulin polymerization promoting protein, or TPPP, and glutamate decarboxylase 2, or GAD2, gene that encodes for the GAD65 protein. They found a clear correlation between the effects of irradiation and lithium treatment on gene expression and the corresponding DNA methylation levels. In addition, looking at figures C, D, G, and H on the right-hand side, this demonstrates how they found that lithium upregulated TPPP and GAD65 levels in the irradiated mice in vivo. This validated the DNA methylations and RNA sequencing data they previously collected. Although there is not a figure on the screen for this, they also found that lithium prevented the irradiation-induced changes in NSPC fate progression observed 4 weeks after its discontinuation. Discontinuation of the treatment promoted neuronal differentiation as well as prevented irradiation-induced astrocyte differentiation. Lastly, we see how lithium ameliorated irradiation-induced spatial learning and memory retention deficits 4 weeks after its discontinuation in figures C, D, and E on the bottom left-hand side. Figure 5C includes heat maps for the first trial of the first day of reversal learning, and then 5D shows the differences in the number of crossings over the previous position of the platform. And 5E showed the time spent in the quadrant where the platform was placed during the previous days of training. Overall, the number of goal crossings was higher for the sham and irradiated lithium mice compared to the sham lithium and irradiated mice, indicating better memory retention in these groups. In conclusion, some things they discussed were that female mice were utilized because they had a higher likelihood to suffer from post-irradiation cognitive defects, but it is assumed that the lithium effects are not dependent on sex, so lithium treatment is likely to aid male subjects to the same extent. In addition, the delayed administration of lithium towards preservation of neurogenesis following whole brain cranial irradiation has never been reported previously. The effects of lithium on tumor cells is only partially explored, so in the future, pharmacological rehabilitation of cognitive late effects after brain tumor treatment may be safer to perform a couple of years after the end of treatment when the risk of tumor relapse is lower. The positive effects of lithium after irradiation are attributed to an epigenetic regulation of genes important for cell fate, maturation, and integration. This is indicated by the increased expression of TPPP and GAD65. The major conclusion from this research article was that pharmacological treatment of cognitive so-called late effects in childhood cancer survival is possible in the future. Thank you.